So, hello everyone. I hope everyone is doing great, and I hope everyone's loved ones are in best health. And today we'll discuss about application of artificial intelligence in biotechnology. As we know, artificial is impacting our life in many positive ways, and in similar way, this artificial intelligence is also impacting sector of biotechnology in huge way, and it is helping mankind unravel many mysteries. So, Sankal, please introduce yourself.
Hello, everyone. My name is Sankalp Jain, and I'm from the Department of Biotechnology.
I'm Sushant Barnawal from the Department of Metallurgical Engineering and Material Sciences.
Hello, Aditya Kachwe. I am from Department of Chemistry.
Yeah, hi everyone. My name is Christopher. I am from the Department of Biotechnology.
So first, let's figure out how biomolecules are there. It's like biomolecules are basically aligned in some particular structure. So it will be like A, C, G, A, T, and it will be repeating itself, and it will be like a different pattern, and it will be basically it's a sequence. So biomolecules are in sequence, and this sequence is like really mystery for humans. This holds huge amount of data in it, and it can explain you about your history, what will happen to you in future, and many other things. It is like mysterious sequence, and we can match this sequence with other sequences to get what is there and what not is there. And basically this right now we use this sequence for figuring out what are functional structure and evolutionary relationship between these sequences. So we can also figure out history from these sequences, and also something about future. Like we can predict, are you going to get some particular disease like ALS or something like that in near future, or we can also figure out like what is your past and what is your genetics, among other things, using these sequences. So these alignments are basically we divide these alignments in two categories. One is a global alignment, another one is local alignment. So what is difference between global and local? So it is very simple. In global alignment, we look at that alignment as a whole. We look at that alignment as a whole. We see that whole alignment. And in local alignment, we zoom on particular part, and we are only concerned about that part only. So we focus on only that small part in local alignment. But in global, we look for whole part. That is, we look for overall alignment, and we try to optimize it. So we compare those two whole alignments, and in local we zoom at particular structure, and we compare just that two small parts of that alignment. So basically methods of we use two methods for this alignment. First one is dot plot, and another one is dynamic programming. So in this dynamic programming, for global alignment we use Needleman Wunsch method, and for local alignment we use Smith Waterman method.
Okay, thank you, Aditya. So, as Aditya said, one of the methods used is the dot plot method. So this is a very basic method where we simply look to uh, place them on one over the other, these sequences, and we try to match if the biomolecules in certain positions are the same for certain sequences. So this is a graphical method, and this was one of the very first methods used when this field was in its beginning stages. So this was initially applied by Gibbs and McIntyre in the 1970s. And we need to put it in context that these sequences are actually millions long. And back then, uh, we had nowhere near the computing power we have today, and nowhere near the storage facilities we had back then. Like no computer had gigabytes of storage in the 1970s. So even for that time, this was considered pretty revolutionary. So the implementation of it is very basic. And as mentioned, it's a graphical method, so it's relatively easy to gain inference from. So what we do is we have one sequence A, which is listed on top of the matrix, and the other sequence, let's call it B, on the vertical side of the matrix, on the left side. So what we do is we start from the first row and we go from the left to the right. And we see that wherever the sequences are matching, we simply put a dot there. For example, here in this matrix, we can see in the first row on the left hand side, we have G. And when we move along the row, wherever the G's are matching, we simply put a dot there. Now, this process is continued throughout the entire two-dimensional matrix, and we just put the dots everywhere. Now, various patterns can arise when we do that. So we can have horizontal lines, we can have vertical lines, we can have normal diagonals, reverse diagonals, we can have X's. And what this representation allows us to do is gain inference from them, which we would normally not be able to do if we looked at it with the naked eye. So um, these are the patterns which we can see. So say if we have a long run of diagonal dots, it implies that these are regions of similarity. Now, if there are multiple subdiagonals, it means that certain genetic sequences are being repeated. Now, these repeated sequences are of huge help to researchers because they help in the process of gene editing. Because repeating sequences can be easily identified and edited. Reverse diagonals indicate inversions, and reverse diagonals, crossing diagonals. For example, in the previous slide, we see that an X is being formed, right? So this X shows that that region is palindromically conserved. Now, again, keeping in mind the context, these sequences are millions long. Now imagine such a huge matrix with so many dots. It would be chaos. There would be so much noise. Like if we get random dots here and there, it shows random matches. So for that, one of the most basic methods used is to simply have a threshold value. For example, we set a threshold value of 6. So meaning that at least 6 dots will have to be together for them to be considered. The rest would be filtered, which would enable us to easily look at the sequences which are bigger and conserved, and yeah, it helps in the inference and understanding process. Now my friend Sushant will help us out with the dynamic programming method.
Okay, so as Christopher has explained about the entire dot plot structure, however, however revolutionary it was quite restricted by the time of its technology. But dynamic programming came, and it was quite evolutionary in terms that we were able to look after large number of sequences. We were able to solve large sequential alignment. It basic dynamic programming basically works into basically has two properties: the optimal substructure, which is just the optimal solution that can be computed from the optimal solutions to the sub problems, and dividing the large problem into several smaller problems called the sub problems. Now these there are several overlapping sub problems such that the total number of distinct sub problems to be solved are relatively small. The one of the major, and the next slide we will find that there is a method known as the Needleman-Wunsch algorithm. Christopher, will you please change it to the next slide? Yeah. So the Needleman-Wunsch algorithm is an algorithm used in bioinformatics to align proteins or nucleotide sequences. It is one of the first applications of dynamic programming to compare biological sequences, and the algorithm was developed by Saul B. Needleman and Christian D. Wunsch. So this algorithm essentially divides a large problem example of full sequence of DNA into smaller series of subproblems, and it uses the solutions of these smaller problems to find an optimal solution to the larger problem. It also sometimes is referred to as the optimal matching algorithm and the global alignment technique. The Needleman-Wunsch algorithm is still widely used for the optimal global alignment, particularly when the quality of global alignment is of the utmost importance. The algorithm assigns a score to every possible alignment, and purpose of the algorithm is to find all possible alignments having the highest score. So in this particular slide, we have a formula where we have a function f of I comma j, which basically determines which basically is dividing the entire sequence present to us into three separate subproblems of f of I minus 1, j minus 1, f of I minus 1 and j, and f of I j minus 1, which corresponds to the diagonal, the column, and the row, respectively. So along with it, we can also find out a special term called s of xi comma yi, which is just a substitution value determining whether there is a match or a mismatch between between two pairs of sequences that we have taken for the process of sequence alignment. The parameter w is just another parameter which we use to determine the gap penalty value in the case of the grid. In the next slide, here we see an example of a grid where we take two sequences TCGCA and TCCA along the row and the column respectively. We start from the lowermost grid square. We start from the lowermost grid square. Over here, we have taken a gap penalty of -2 and a match score of 1 and a mismatch score of -1. So yeah, here, which means over here the substitution value s of I xi comma yi is -1 for a mismatch, 1 for match, and the parameter w is -2 in this case. We start from the lowermost square. Uh, you can One more important insight is what we can see over here is when you see the topmost row and the leftmost column, there's an incremental value as you go towards the right. For example, in the row, as you go towards the right, there's an decremental value of -2. This shows the gap value that we have put into each of the squares as an initialization step. To solve the problem, we start from the lowermost area, and as we see the path, as we travel through the path as shown by the red arrow, we have to reach the most optimal position. That's the 0, 0. After solving for this entire grid, we get as the two sequences shown, we can see that the most optimal score is 2 as obtained by the sequence matching of these two algorithms, by sequence matching of these two DNA sequences. So the another uh, example is will be carried on by my friend Sankalp.
Okay, as Sushant explained, the Needleman-Wunsch algorithm, which was used for the global alignment. So now we'll discuss the Smith-Waterman algorithm, which is used for the local alignment. So Smith-Waterman algorithm performs local sequence alignment, that is for determining similar regions between two strings of either nucleic acid, that is DNA, or protein sequences. So here we will look into DNA sequences. So instead of looking at the entire sequence, the Smith-Waterman algorithm compares segments of all possible lengths and optimizes the similarity measure. The algorithm was first proposed by Temple F. Smith and Michael S. Waterman in 1981. So, like the Needleman-Wunsch algorithm, it is also a dynamic programming algorithm. And as such, it has the desirable property that it is guaranteed to find the optimal local alignment with respect to the scoring system being used, which includes the substitution score and the gap scoring scheme. So the main difference to the Needleman-Wunsch algorithm is that negative scoring metric cells are set to 0, which renders the local alignments visible. So once we fill the entire matrix, the traceback procedure starts at the highest scoring matrix cell and proceeds until a cell with a 0 is encountered. But in Needleman-Wunsch, we like traced back till the 0, 0 itself. So let's take a look at the example, how it is done. So here we are taking two sequences. So first one is ATCGAA and second sequence is CATAC. So as we can see that in the Needleman-Wunsch algorithm, there were negative scores which were for gap penalties. So here gap penalty is -4, match score is 5, and mismatch score is -3. So we can see in the matrix that there are no negative scores because we are setting all the negative scores to 0. And finally, when we are filling it in the similar fashion as we did in the Needleman-Wunsch algorithm. So we are dividing it into sub problems, and we are calculating the current matrix based on the calculated sub problems previously. So once the entire matrix is filled, uh, we have to look for the highest scoring cell in the entire matrix. So here in our example, the highest number is 11. So that will be the optimal score for our local alignment. And now we have to trace back from this cell to the first zero we encounter. So as we can see that we are tracing back from 6 to 11 to 6 to 10 to 5 to 0. So here we'll stop. So finally, our optimal local alignment will be ATC. So ATC will be our optimal local alignment. So this is the complete example. So now Christopher will conclude the whole thing.
Thank you, Sankar. So, thank you for your time. I hope we were able to give you a glimpse into this field of sequence alignment. Now, this field is a very happening field, especially due to the advances in machine learning techniques. So, as we presented to you three algorithms: the dot matrix algorithm, and the other two dynamic programming algorithms. So nowadays, there are neural networks which can do what these algorithms did much more quicker and much more efficiently as well. And the impact of this field has been huge. Researchers able to find out which organisms have a common ancestor. Researchers have been able to figure out which sequences are like precursors for certain disorders a person might face. And this has helped in the efficient treatment of such people. And the list goes on. I hope we were able to give you a small glimpse into this field. Uh, yeah, thank you, everyone. Thank you for your time.